Good afternoon, everyone, and I'd like to begin by thanking Geraint very much indeed for giving me the opportunity to speak to you this afternoon for 20 minutes to tell you about the research that we're currently doing in Durham and my particular area of greatest scientific excitement. So I'll be talking today about decoding calcium signaling in plants. And what I want to do is really just to take you through this work in the form of 4 questions. And start off with, just by way of introduction, although most of you will know what calcium signaling is, I thought I'd give you a light introduction into what calcium signaling is. Then lead into what the problem is. So we know quite a lot about calcium signaling, so what is the problem? And by problem, I mean on 2 different levels. The first level is, what don't we know about calcium signaling? What do we need to understand and solve currently? And secondly, I mean, what is the problem contextually for a plant in terms of having to make sense of the environment and taking external information and being able to process that and make a correct, informed decision without having a brain? So this is on 2 levels, is what is the problem of calcium signaling? The third thing I'd like to do is to, obviously I'm talking about decoding of calcium signaling in plants, so I'd need to show you some form of evidence to convince you that calcium does actually encode information in plants, is used by plants to interpret environmental signals and cues in order to be able to produce an end response that is appropriate to those cues. And then finally, I want to tell you a little bit about what we know about how plants actually decode these signals. So this work is in its infancy, but we're starting to get to a point where we can start to decipher the cellular language that is used by plants to communicate internally to result in the correct response to the environment. So first of all, what is calcium signaling? Well, calcium signaling is a universal phenomenon in biology, and in fact, you're currently hopefully using calcium signaling to either listen or to look at my slides. And the idea is that stimulation will lead to changes in concentration of calcium. So this ionic cellular concentrations of the cells change, and this leads to an end response to something happening. So in your case, you could be seeing things or hearing things, or you could be sensing touch. In the case of plants, we're particularly interested in how plants will respond to environmental cues, things such as light, temperature, and so forth, and to lead to the correct response that they need to coordinate growth and development. And my particular interest, or the interest of our lab, has been for the last couple of decades, is really beyond how plants regulate the expression of specific genes that they are required to combat stress. So abiotic and biotic stress will lead to changes in gene expression, and calcium is instrumental in playing the sort of middle manager part to convey that information from the stress leading to the nucleus and gene expression. And of course, gene expression is very important for plants. They can't move. They need to be able to reconfigure themselves to produce stress proteins in order to be fit to be able to survive the stress itself. And so they need to be able to activate correct complement of proteins to allow there to be tolerance to particular stress. So that's calcium signaling. We can show that in plants. If you see from the previous slide saying that calcium is important, it's very well for me to say that calcium is important for mediating stress gene expression and tolerance. But there is a lot of evidence in the literature, hundreds if not thousands of papers showing that calcium is absolutely necessary and in some cases sufficient for mediating stress tolerance, a whole variety of stresses. Here you see a picture of a calcium response in some Arabidopsis seedlings responding to osmotic shock. So in other words, drought stress mimicked in the lab. And the bright colors you're seeing here, just us measuring calcium increases underneath the camera. You don't need to worry about the technology that we use here. And what you can see is, if you look at this in terms of temporal response, here's the calcium concentration in the cells going on the y-axis and time on the x-axis. When we mimic drought stress by applying an osmotic shock in the form of mannitol, we see this lovely discrete transient of calcium. Now, how do we interpret this? How can we find what this calcium is actually doing? Well, we have a number of ways in which we can block this response. So we can use things called calcium channel blockers, such as lanthanum, which in the presence of the stress, will limit the amount of calcium that increases in the cells. In other words, we can see what is the consequence of allowing the response to occur, but to block the calcium specifically. And this is very old work, but if you look at the expression of a given gene, and this is a gene, P5CS, which is important in combating osmotic stress because the gene product here is important in re-establishing osmotic balance. You can see under the two conditions of no treatment, there's very little expression of this gene. This is transcript levels, you can see here, represented by the brightness of this band. When we stimulate osmotic shock, we get an increase in expression. But in the presence of lanthanum, where we got this reduced calcium elevation, we also get reduced gene expression. So this is showing that the calcium, these pretty calcium elevations in response to drought stress, are absolutely necessary to mediate the expression of at least one gene, which we know is important for protecting plants against drought stress. Now, one gene may not actually contribute hugely to stress tolerance, but is a component. But this picture, sorry, I should have said over here, of one gene being regulated by calcium is actually manifold, and there are hundreds of genes in different stress responses that are controlled by calcium. So together, those regulated genes will lead to an effective difference in the quantitative stress tolerance of a plant. And here's another example. So this is now not measuring gene expression, but measuring physiological tolerance to a particular stress. This is pathogen stress, and in this paper with Murray Grant way back, we showed that avirulent Pseudomonas caused an increase in calcium response, which was needed for the tolerance response to that avirulent pathogen. And what you can see in the top panel as the control is half a leaf of Arabidopsis being infiltrated by this pathogen, and it looks sorry for itself, but actually this is a winner, because what it's actually doing is to prevent the pathogen from spreading by causing localized cell death. Now, what we can see is that if we prevent the calcium elevation that we know occurs when these cells are seeing the pathogen, again using lanthanum to block the calcium response, the leaf looks normal. There's no cell death, which you may think looks like a healthy plant, but actually what's happening here is this plant has not been able to manifest the correct response to combat the pathogen, and so the pathogen is free now to move into the rest of the plant. So this is showing that stress tolerance, quantitative stress tolerance, is often regulated by calcium, and calcium is necessary for those. So these are two examples. So what's the problem? Well, if we look at this from an evolutionary perspective, as well as a scientific perspective, this is the problem. If we attack plants, or plants are attacked in the wild by various different stresses, such as drought, cold, and pathogens, I've already talked about drought and pathogens. Here's cold in the middle here. Calcium will be elevated in cells in the plant. And indeed, this next diagram shows you all different types of signals, not only stress signals, but internal, endogenous, hormonal signals, and so forth, which will cause increases in calcium. So calcium is effectively involved in hundreds of different primary signals. Now, the problem with that is it's a problem of interpretation. How calcium is calcium is calcium. It's not like a family of proteins where there's different forms. How can an elevation in calcium convey specific information? Because obviously the response to cold, the response to drought, the response to pathogens, the response to different hormones are necessarily needed to be different. The outcome needs to be different, yet they're all causing an increase in calcium. So this is a problem that we are very interested in academically, so that's where the problem comes from. How do plants deal with this problem? But also, in terms of evolution, it's a problem that plants have to deal with, because we don't have this problem, because all our different signals are nicely packaged, because we have specific receptors for all the different primary signals, and we have specific neurons which will inform the brain of what that signal is. So we don't need to worry about this so much. But in the case of plants, they don't have a brain. They don't have specialized organs to actually process information. So they need to have a way in which they can use calcium to still maintain the faithful primary signal information. So and I emphasize, one possible way in which calcium can convey information, specific information, is through the concept of calcium signatures. And there are other ways, and I'm sure people will ask me questions about this, but there are other ways in which this can occur. And the idea of calcium signatures is literally that yes, different signals will cause an increase in calcium in cells, but those increases are not the same. They show temporal and kinetic differences between them. So in other words, if you look at cold here, you have a biphasic calcium elevation, drought a monophasic, and things like nod factors will cause oscillations. Now you as a human being can look at these and you can tell them apart. And the question is, can plants tell those apart? Because if they can, then they have a system, they have the ability, therefore, to maintain the primary specific information of a primary signal and encode it into a calcium signature, which is what these are, and then they can then decode it to give the appropriate response. So what is the evidence that calcium actually does encode information in plants? So if I'm being harsh, I'm saying, well, we observe that there are different calcium signatures at different primary stresses, but that doesn't mean that the signatures are actually used. That's just an observation, it's correlation. So to get an idea of whether or not plants could actually see calcium signatures and decipher them and see them as different, we gave plants presented them with artificial calcium elevations. And what you're seeing here are oscillations of calcium. They're real increases in calcium measured from real plants, but we have basically tailored the responses by stimulating plants with electrical impulse to generate a calcium signature at will. So we're creating here, in this particular case, different oscillations of calcium. And the idea is to present these to plants and say to the plants, can you tell the difference? And here's an example. So this is a nice simple example. There are 3 different calcium signatures here. They are all oscillations and they all have 10 spikes. What you can see is the oscillation here in part A has relatively high amplitude compared to the other 2 oscillations. So these 2 have lower amplitude, so they share a parameter of low amplitude, and this has a different parameter of high amplitude. These 2 have a higher frequency than this one. So the peaks appear twice as quickly as they do in this one. So these 2 share the property of frequency. So we asked the question, can plants tell these apart by applying these to plants and measuring the expression of genes in those plants using transcriptomic analysis? And the next slide shows you what the outcome of this is. So those 3 signatures, 1, 2, and 3, what you're seeing here on the Venn diagram on the left-hand side is how many genes go up in response, or the expression is increased in response to those signatures, and on the left-hand side, how many go down, or decreased. But importantly, if you look at the numbers, the most important thing about this slide is that it shows you there's relatively little overlap. So signature 1, signature 2, and signature 3 will increase the expression or decrease the expression of largely different genes. So this would suggest that plants can tell those different signatures apart, and the result of that is to induce the expression of different genes. And if you can investigate this a little further, if you take the genes that are induced by signature 1, signature 2, and signature 3 and analyze the promoters, what you find is there is a preponderance of different promoter elements in those 3 different sets of genes. In other words, it seems likely that the different signatures are activating different transcription factors. So we have the raw materials there of the signature encoding information which can be interpreted and activate different transcription factors to lead to different gene expression. So it seems that this system is certainly likely that it can operate in plants. Now, obviously, this is an artificial situation where we're coming along and presenting the signatures to plants. Excuse me. Does this actually occur in nature? And there are a lot of examples of this in more natural conditions. So this is just one example, a recent one from the lab here from PhD student Bridie Jacobs. And what you can see here is what happens to the calcium concentration in Arabidopsis seedlings when we challenge them either with mannitol to mimic drought, and you can see this calcium signature is predominantly a rapid transient and then it flatlines, or the chemical signal, Flagellin peptide signal, which is a PAMP, which is initiates the primary plant immune response, where you get really little primary initial transient, but you get this characteristic transient, prolonged transient later on, so prolonged response, second peak. And to cut a long story short, for drought responses, this rapid early transient is important, and for the pathogen response, it's this later one that's important. And what happens when you put these two together? If you put these two together, what you can see is that the pathogen second wave, if you like, is enhanced, whereas the first wave, which is the mannitol-induced wave, is reduced. So to us, if you look at this, the calcium signature suggests that the plant is favoring, is seeing, okay, in response to mannitol, it will respond and induce the right response. In response to flagellin, it will induce the right response. They're different responses. But when the two come together, the plant is favoring the defense response over the drought response. And if we look at this in terms of the outcome, so here's a gene called KIN2, which is a drought-induced response that's regulated by that calcium signature. And here we have the control conditions of water, where you have no expression. Treat them with mannitol, and this corresponds to the red trace you saw in the previous slide. You see you get induction of gene expression. So this is the plant trying to combat the osmotic stress. However, in the combination of flagellin with the mannitol, and you remember the flagellin seems to be, in combination with the mannitol, seems to attenuate what we think is the important part of the calcium signature for the drought stress response, you do indeed attenuate the downstream consequences of that. So the plant is physiologically favoring defense against the pathogen that it thinks it's seeing over the drought stress response. So we think that this concept of calcium signatures is operating to regulate specific gene expression and making decisions in plants leading to change in stress-induced gene expression. Okay, so we get to the final part, which is really the key thing: is how do plants decode calcium signals? So if we just simplistically break this down to what do we need to be able to decode calcium signals? So at the top here, I've already described to you how calcium signals are encoded. A specific stimulus will lead to a calcium signature which is characteristic for that stimulus, and that's the encoding part. Now, to decode, you need at least, to lead to expression of genes, you need to have at least two components, extra components. There may be more in some cases, but you need at least two conceptually. The first is a calcium binding protein, which can bind the calcium that's increased in response to this and act as a decoder. And the second is at least one transcription factor. And that's the decoding apparatus. So we have focused on transcription factors called CAMTAs. And these are transcription factors, there's 6 of them in Arabidopsis, that bind a calcium binding protein called calmodulin. And each calmodulin binds 4 calcium ions. So in other words, the active component here, the transcription factor with calmodulin and calcium, which will bind and regulate gene expression, has 6 components, 4 calciums and 2 proteins. Now, this is the situation. This looks complicated because it is. This is the situation in the cell of how you get to produce that component, that 6 component complex. What we have here is each of the points at the end of these arrows is an incomplete version of that 6 component complex. So if you look on here, this is an example. You can have calmodulin with just 2 ions here, calcium ions, 2 calcium ions in a different place, the proteins together, but only 2 calcium ions on the calmodulin and so forth. So each of these points at the end of the arrows is one of those. There's 18 possible forms. And the R here, the arrows in between, are the interconversion between those based on basic biochemical thermodynamic principles. Now, the key thing is, what we're really interested in is how much of this is produced at any given time. Now, that will change. How much of that is produced, is present in the cell, will change depending on all these interactions, and those interactions will all change depending on what calcium signature comes into the system. Now, obviously, you can see this by eye, that we can't compute this in our brains. It's not possible. So the bad news is that we have to invoke mathematics. And to use mathematics to be able to model this, to be able to allow to make a prediction based on this as to what a calcium signature will do, in a specific calcium signature will do in terms of the production of this final complex and thus gene expression. And this is indeed what we've done. So for instance, if you take 3 calcium signatures which occur in nature, a single transient, a prolonged response, or an oscillation, and then you look at what this should do to the expression of genes regulated. My timer's going, don't worry, I will only be another couple of minutes. What will happen to the expression of genes regulated by the CAMTAs, you'll see that the oscillation here greatly induces the expression of genes, but the other 2 are largely ineffective. So the transients and the prolonged response does not seem to invoke this transcription factor to wake it up to induce gene expression. Excuse me. But the oscillation specifically is able to activate expression. Now, one very interesting thing about this is that this is done with the computing, using a KD, i.e. the affinity for the transcription factor to the DNA of this level. If we change that, this just show you what the palette of nature has to tweak this system. If we change that by an order of magnitude, so change the affinity of the DNA and transcription factor by an order of magnitude, you can see now the transient becomes the most potent calcium signature for inducing gene expression. And in fact, the oscillation is still potent, but the prolonged still doesn't have an effect. So this system, this response is nonlinear, and it contains a large number of parameters which nature has tweaked over the millions of years of evolution to allow specific control, specific transcription factors by specific calcium signatures. So it just remains for me just to finish by giving you the very broad conclusions and future work and thanking a few people. So the take-home message is that calcium signaling is involved in many, many different stimuli, pretty much everything that plant sees and has to respond to. It acts, and this is one of the thing we're most interested in, to regulate stress gene expression, which leads to tolerance. And hopefully you've got some measure of this and believe this, that specific genes are activated under different conditions by the use of calcium signatures. So the external information is converted into an internal signal, which in the form of a calcium signature, which then in turn needs to be decoded by the plant cells to lead to the correct responses. And this requires at least one calcium binding protein, at least one specific transcription factor. Now, we've had to combine modeling, mathematical modeling, and empirical analysis to allow us to understand this mechanism. But this is starting to allow us to make predictions as to how the decoding, to be able to make predictions as what the outcome will be for a given calcium signature. But more importantly, it's giving us information about the mechanism. And currently, we've been working on CAMTA and CpG60G transcription factors and calmodulin and the plant immunity response. But in future, we're hoping to extend this to other transcription factors and other calcium binding proteins and other biological contexts, as well as plant immunity. So I just want to thank the people who were involved in the work I've just presented today. Helen Whaley, the PhD student who was involved in transcriptomic promoter element work and the calcium signatures, the artificial ones. Bridie Jacobs was involved in the work on the natural calcium signatures in combined stress. And a big shout out to my colleague, very clever man, Junli Liu, who's the person who's doing the mathematical modeling. Obviously, I didn't do that myself. And I'd just like to thank you all very much indeed for listening out there in cyberspace. So thank you very much.
Thanks very much, Mark. It's very interesting indeed, and well kept to time, I think. So we have a few questions. So first one is involved with the model there you were talking about. So this is from Hohan Go, and he asked whether the model fits the experimental observations that you.
Yes, I didn't have time to. Geraint's frozen. I hope it's not. Have I frozen as well? Can you hear me?
No, you're okay. You're back now.
Okay. That's a very good question.
Yes,
indeed. I just didn't have time. So to show that we have actually, they do fit for the plant immunity response. They fit the observations definitely. For the natural signals, we had the example I gave you there for combination is just one of many where it fits. So it does indeed, yes.
Okay, so a question from a speaker we had a couple of weeks ago. So Carmen Escudero Martinez, she asks, why do you think the plants appear to prioritize biotic stress over the abiotic stress in the example that you gave?
That I wouldn't be able to tell you. because I'd imagine it's depends. So I should say we're using young seedlings for these experiments. So the prioritization may be different at different stages of development. But why that is the case, I don't know. If it, I wouldn't be able to tell you, to be honest.
Okay. So a question from Pavithra Narayara, quite a long question. So he talks about the proteins that bind to calcium, like calmodulins and
Yes.
CPLs, and they're referred to as calcium sensors.
Yes.
But not all these proteins have been shown to sense calcium. So do you think there needs to be a discrimination between calcium binding proteins and calcium sensors?
I'm not sure what that refers to. I mean, basically all proteins that bind calcium have the potential to sense. I mean, as long as they can transduce that sensing event to a downstream consequence, then I think they all have the capacity to do that. I'm not really sure quite what the question means.
Yeah, I'm not sure. As everyone should know, so all the questions I'll pass on to Mark afterwards.
Okay.
And so, if
I mean, maybe one way to answer this is that for a calcium sensor to be a sensor, it needs to operate in a certain range of calcium concentrations. There are a lot of plant calcium binding proteins which are operating outside of those ranges, which may be used to sequester calcium, for instance, and so forth. So there may be some calcium binding proteins which are not sensors in the strictest sense of the word. They could be used as capacitors for stores and things like this. Definitely.
Okay. So that kind of fits with the next question. So this is from Arjan Barman, and they ask, how are the different concentrations of calcium generated within the cell? Is there any correlation to the events that raise the concentration?
How are the concentrations raised? So basically calcium kept very low in the cytosol. The plant cells work very hard at this by pumping calcium out, either outside of the cell into the cell wall, or the apoplast rather, or into organelles. And then to increase the calcium, that's reversed by proteins called calcium channels, which allow the calcium in. And what was the second part of the question?
Hang on, where are we? No, I think that was it. So just the correlation between events that raise the concentration.
Yeah, I mean, I think what the question may be asking there is there the primary signal has to regulate to cause a different calcium signature. Does it do this by regulating different parts of the calcium machinery? And the answer would be yes. So for instance, there are certain signals such as cold, which primarily the calcium all comes from outside the cell, and there's other signals such as touch, for instance, which we think comes primarily from inside. And there again, there's also another level of specificity. It could be that rather than measuring the signature, in those cases, it's possible that the cell is also measuring where the calcium is coming from. I think maybe that's where the question is coming from, and that is important.
Yeah,
that
maybe
this
question maybe along the similar lines. This is from Brand Graff. He asks, as there are multiple abiotic and biotic stresses coming in at any time, how does the plant deal with all the different things on the cellular level? If there's multiple signals coming in.
So I think so. This is a limitation of the current work, ours included, is that we're kind of averaging our responses. So if you think about this, that there could be calcium responses in different cell types, and within the cell there could be calcium responses that are limited to discrete places in the cell, for instance. So there's another level at which the calcium signals could be discriminated by either the plant, if it's different cell types, or the cell itself from where it's coming from. So I think that fits in that very well, and that's definitely something we need to start to overlay into the model to increase that complexity. So that's absolutely right. And indeed, in animal systems, there's very good evidence. For instance, if you have calcium elevating near mitochondria, you get one response. If you have calcium elevating near another organelle, you get different response. So the cells have the capacity to sense microdomains of calcium where this calcium is coming from. So that is a very important point. And on top of that, you've got the differences in different cell types as well. So that's a very good point.
Okay, so we have a question from Monica Garcia, who asks about the legacy. So do you think, with previous exposures to calcium, does that have an effect on the signature of future responses?
Yeah, lovely question, because I'm happy someone asked me that. Yes, very much so. We're seeing this time and time again, that not only from the same stress followed by the same stress another time, so acclimation, but between stresses. So I showed you the example here of combined stresses, which were applied at the same time temporally. But if you apply a gap between them, you do see a memory effect. And the effect isn't necessarily always the same. So the amount of time that elapses between them will affect what the outcome of that is. So the answer is absolutely yes. But the key question to that is, how is that memory preserved? And that is a big question, and we have absolutely no idea. One could imagine, going back to the previous question, that it's somehow the calcium transporters which are creating the calcium signatures are themselves modified permanently in some way, which allows the memory to be registered for future events. But the answer to that question in short, which I didn't do in short, I did very long, is yes.
Okay, so a final question. I got to thank everyone. We've had loads of questions coming in. Actually, it's been by far the most questions we've had for any talk. So you obviously stimulated some interest. So as I said, I'll pass these on to Mark afterwards.
That'd be great. Yeah.
Yeah. The final question though. So from Anna Laxtel, they ask, what do we know about calcium concentrations and regulation within the nucleus?
Okay, so my view on this is that the nucleus, certainly when we've measured nuclear calcium concentrations, they are very, very similar to what happens in the cytosol, and we see very little difference. However, many groups are reporting sort of independent nuclear responses, so separately. So this is also a very good, the question the person asked that is a very good, made a good point, because obviously we're looking at transcription factors and the interaction with transcription factors which actually are in the nucleus. So in our hands, in our lab, we've seen very little difference between what happens in the nucleoplasm and the cytoplasm. So the signatures we are measuring are the same, but there are instances in the literature definitely where they are different. So there's room for both.
Okay, great. All right, so thank you very much, Mark, and that was a really interesting talk and stimulated.
Thank you very much. Well, thank you very much for inviting me and everyone for listening and asking such good questions. Appreciate it.
No worries.